Our immune systems produce antibodies in response to germs. But in lab, we can produce antibodies by reprogramming cells to make them. Now, why make antibodies if our immune system already does this? Well, these synthetic antibodies are used in immunotherapy, a treatment that mimics how our body fights disease. Antibodies specifically target disease cells. We can attach drugs to antibodies to deliver the drugs directly to the site of infection. This powerful treatment is used to treat many diseases, like cancer, Alzheimer's, and arthritis. Unfortunately, though, we can't program cells to just make antibodies. They also make many impurities. These impurities cause undesirable side effects in patients and should be removed from our medicines. And that is where my research comes in. Antibodies, like the guy on screen, are large molecules made up of tiny amino acid building blocks. Now, some impurities differ from the antibody by as little as one amino acid out of hundreds. So removing these impurities is necessary and important, but also very challenging. Companies spend many months and many millions testing hundreds of filters and conditions to purify new antibody drugs. They even use robots to do this mountain of experiments. Our lab's goal is to help companies purify new antibody medicines better, faster, and cheaper. My research will help guide filter selection. I study interactions between filters and antibody fragments called Fabs. As a model system, our lab made 40 antibody fragments, or Fabs, that differ by 1 or 2 amino acids. First, we did a bunch of experiments. We collected data for the antibody fragments on many different filters. Using this data, I developed models to predict how a new antibody fragment would interact with filters before we do any experiments. I used the models to study how Fabs with such minute differences could have such dramatically different interactions with the filters. So, to purify a new antibody fragment from a very similar impurity, you can plug this Fab into my models and test the filters in minutes on the computer. Then, you only have to do a few experiments in lab based on the model predictions. So my research helps engineer the purification of new antibody medicines. These models can help companies purify new antibody drugs better, faster, and cheaper. This can also help patients get access to these drugs more quickly after they're discovered. This helps make antibodies more accessible to everybody. Thank you.